Thank you very much. I'm happy to be here, and it's nice to see familiar faces out there. In the interest of lowering expectations, I have to say that this is the first time that I've ever given a talk without a PowerPoint presentation, because that's what we do in science, is we have lots of pictures to kind of divert your attention to what we're saying. So thank you for having me here. I'm honored that the Center for Dominican Studies asked me to speak to you today about truth in science, and I will do my best. I will focus my remarks specifically on the branch of science of biology, or the science of life. From St. Albert the Great to the Sisters in Science at Ohio Dominican University, all have pursued and contemplated truth in science and shared the fruits of those efforts with others, seekers and teachers of the truth. From the beginning of our human existence, our minds have asked the questions, what, why, how, and even who. Ancient humans looked at the stars and thought they must be distant campfires. A reasonable explanation around that campfire. 300 years before the birth of Christ, Aristotle assigned forms of life into categories and hierarchies and created what he called the ladder of life. Organization, another apparent intellectual drive of humans. The so-called father of microbiology, Anton von Leeuwenhoek, ground magnifying lenses to inspect the quality of textiles in his trade as a Dutch draper in the 1660s. As boys will be boys, or more aptly, humans will be humans, he soon turned his lenses on blood, sperm, and pond water. He made some of the first observations of a previously unknown and unseen microscopic world. He named the fantastic pond creatures animalcules. Can you imagine talk about being impressed with the world? Can you imagine seeing an unseen dimension that no one had ever seen before, and it was full of life, both around us and even in us? At about the same time, Englishman Robert Hooke turned his lenses to the bark of a tree and saw uniform squares composing cork, and he named them cells, reminding him of small cubicles occupied by monks. By 1839, German biologists Matthew Schleiden and Theodor Swan had established a basic tenet of what became cell theory, based on supporting observations made by themselves and many others, that all living things were composed of cells. Later, the idea that all cells come from pre-existing cells completed cell theory, which is one of the foundations of biology. It is also a great example of discovery-based science. A word about the word theory. This word has a different meaning in science versus how it's used in everyday life. In science, a theory is a body of knowledge, long-standing, always supported, and never refuted. It carries the weight of a law. In everyday usage, it's quite different. It means more of a guess or a conjecture. Very different meanings, and the root of not a little misunderstanding. Back to cell theory. All cells come from pre-existing cells. It seems like a self-evident truth. But for several millennia, reaching back to Aristotle, some forms of life were believed to spring spontaneously from non-living matter. The idea of spontaneous generation persisted until the last half of the 19th century. Without broad understanding of the microscopic world, it was believed that life could spring from non-life, that mice sprang from dirty clothes, that flies were generated de novo from rotten meat. It was what your eyes told you. And where do babies come from? Yeah, you're all old enough. Okay. It was thought human sperm contained very tiny, miniature, fully formed humans called homunculi, and if mixed with cow manure for nutrition, babies would spring forth. Perhaps that says something about the status of women at the time, too. So how did the truth come to light? The answers came from experimental science over several hundred years. In introductory biology, we talk about the challenges to spontaneous generation, beginning with the experiments of Francesco Redi in 1668, who investigated the origin of maggots on rotten meat. He took 2 portions of rotten meat, he left one open to the air and everything in the air, and the other he covered with a fine lace. You guessed it, no maggots on the lace-covered meat. A score against spontaneous generation. In 1745, John Needham boiled meat broth and immediately sealed the containers. The broth still clouded up, indicating growth, supporting the idea of life from non-life. Lazzaro Spallanzani in 1768 revisited Needham's experiment, but introduced a partial vacuum into containers. No growth, but he had introduced another variable by removing air from the experiment. The controversy continued. Finally, in 1859, Louis Pasteur, another father of microbiology has more fathers than the Maury Povich show, by the way, it seems like, performed the famous Swan neck experiment. He used glass flasks with curved necks. This is where the pictures come in, see, we don't have to, with curved necks, like a goose or a swan. This allowed air to freely pass in and out of the flask, but the swan part of the curved part of the neck created a trap for any particulate matter. One flask he left with the swan neck intact, and the other flask he broke off so that there was a direct opening to the air and also to any particulate matter, like dust carrying microbes. That flask became cloudy, and the swan-necked flask stayed clear. It was the experiment that killed the idea of spontaneous generation. It is believed that the controversy persisted for so many years due to the unknown, at the time, nature of bacterial spores. Bacterial spores are dormant, protected forms of some bacteria that are resistant to killing by the usual sterilization methods of boiling. Over the years, conflicting results of experiments due to sporulation prolonged the discussion. In the end, cell theory prevailed, and today seems self-evident. All living things are composed of cells, and all cells come from pre-existing cells. Another major example of establishing truth in science is the germ theory of disease. Robert Koch, a German physician and known as the father of modern microbiology, more paternity involved here, established a relationship between bacteria and the diseases they caused. Previously, illnesses were thought to have been caused by evil spirits or were inflicted as punishment for sins. Some diseases were thought to be caused by miasmas or bad air. I like to read memoirs of pioneers who settled on the Midwestern prairies, and they speak of situating their homes off of the prairie inside of a tree line in the woods to avoid prairie fires, but not too far in the woods because they felt that the bad air would cause illness. Incidentally, Charles Darwin wrote of miasmas in describing some of the areas in South America, these stagnant ponds and such. Anyway, Robert Koch's quest for the true cause of disease began with establishing methods for the isolation of pure bacterial cultures in the 1880s. It is still today the important initial step of characterization of any microbial culture. He established what have become known as Koch's postulates and the germ theory of disease. There are 4 simple steps, revolutionary at the time, for establishing a causal relationship between specific isolated microbes and the resulting disease. Number 1, the microorganism must be present in every case of the disease and absent from healthy individuals. Makes sense. Number 2, the putative causative agent must be isolated and grown in pure culture. Number 3, the same disease must result when the cultured microorganism is used to infect a healthy host. And 4, the same microorganism must be isolated again from the diseased host. Now, before you kind of freak out and think, what was he doing going around infecting people with all sorts of stuff, he actually used sheep and a mice model to do this. His initial work was on Bacillus anthracis, the causative agent of anthrax. And he also studied cholera and was awarded the 1905 Nobel Prize for Physiology and Medicine for his work on tuberculosis. Much of science is descriptive or discovery-based. For centuries, we have cataloged and described animals and plants, and today we still study life in caves or on islands. Take the beautiful artistic depictions of birds by Audubon or the wonderful detailed anatomical drawings of Leonardo da Vinci, examples of observation and documentation of the natural world at its finest. With the advent of DNA sequencing, description has become molecular. DNA sequencing data can reveal the causes of genetic diseases, the parentage of an offspring, or the very genetic code of a fluorescent protein from a jellyfish that can be cloned into any organism. DNA sequencing can reveal the true phylogenetic trees or relatedness of organisms that in some cases are very different than those inferred by the outward morphology. True general principles in science are pursued by hypothesis-driven investigation or by use of the so-called scientific method. This process is said to be iterative and self-correcting in the following way. In these steps, the first step is to make an observation of interest, and I'll talk about an example after this. And the second step is to formulate a possible informed explanation for your observation, a so-called hypothesis that explains it. You use the hypothesis to make predictions, and you test those predictions by experiment and reject, accept, or modify the hypothesis according to results. You repeat steps 3 and 4 until the experimental results reflect a true hypothesis. Notice I said if the experimental results do not support the prediction of the current hypothesis, it is modified or rejected. Hypotheses that are consistently supported become theories and must stand the tests of time. They are never proven per se, but remain open to modification if the scientific results require it. The best known example of the use of the scientific method is that of 19th century naturalist Charles Darwin and his findings published in the book On the Origin of Species by Means of Natural Selection. Many people at the time believed that all forms of life were created in a single creation event and that life remained unchanged since. However, others shared the view that Darwin held, that life changed over time. What distinguished Charles Darwin was that he proposed that the mechanism that drives evolution, natural selection, was the key to his take on evolution. It's not just survival of the fittest, but that they reproduce more. It began with an amazing opportunity for Darwin to make his observations. In 1831, at the tender age of 22, he signed on to a 5-year voyage on a tiny ship, the HMS Beagle. And it was tiny, 90 feet. 5 years on a 90-foot boat with all your best friends. Oh boy. So as they sailed around the world, he actually suffered from seasickness and spent all the time he possibly could off the ship and taking notes on the life that he observed on land. As they sailed around the world, he noticed variation in similar species depending upon geography. His famous example, the beaks of the Galapagos finches. He proposed the ancestor of these finches came from the South American mainland a couple of million years before, and as the birds spread across the island archipelago, they evolved specific beak shapes most suited to the food source on each island. He called it descent with modification. Darwin then asked the question. He already knew that species could be shaped by observing the artificial breeding of varieties of dogs and pigeons. Darwin was also aware of Thomas Malthus's essay on population, stating that more organisms are always born than survive. And Darwin asked the all-important question, why do some survive and others do not? He hypothesized that the organisms most fit for their environment would survive to reproduce more. Natural selection. He wrote the manuscript in 1842 and threw it in a drawer. There's some family things going on there that I'm not going to talk about, but he came from a very conservative family. When Alfred Wallace arrived at the same hypothesis as Darwin in 1858, they decided to present their work together. Over the years, the predictions that flowed from the natural selection hypothesis have been solidly supported. Classically, we talk about 5 lines of evidence that support the theory of evolution. The first one is the fossil record, and this one has as time has gone on, virtually all the transitional forms of fossils or missing links that you might think of have been found at the predicted intervals of time. Also, microscopic fossils dating back to 3.5 billion years ago have been discovered. The second line of evidence is the age of Earth. Based on radioactive decay, the Earth is 4.5 billion, B with a billion, years old. It is this unimaginable expanse of time that makes natural selection and descent with modification from an ancestral cell feasible. The third line of evidence, and this is all things that we talk about incidentally in our introductory biology classes. The mechanism of heredity. About the same time that Darwin was making his observations, Gregor Mendel, the Austrian monk, was working out the biological basis of heredity, while Darwin was looking at the broader implications of these genetics. Number 4, comparative anatomy. Studies of anatomical structures reveal the relatedness or homology of certain structures such as vertebrate forelimbs. Humans, cats, bats, horses, porpoises, we all have homologous parts in or similar parts that have changed just in various degrees in our forms, for example. And 5, the last line of evidence is molecular evidence. We're now able to look at the relatedness of organisms at the molecular level. The amino acid sequence of evolutionarily conserved proteins, for example, demonstrate the branches that we share on the tree of life. The more highly shared or conserved features of biochemistry indicate the more ancestral the trait. For example, all living things on this earth use the same 4 bases in DNA sequence, A's, T's, C's, and G's. It is universal. I mentioned Darwin wrote the preliminary draft of Ideas of Evolution by Means of Natural Selection in 1842 and put it in a drawer for 16 years. Clearly, Charles was not pursuing tenure and promotion. Publication was then and still is the method of sharing truth in science. The scientific information we accumulate is archival. No one scientist starts from the beginning, but builds on what has been established before. Scientific knowledge as an archive, then, must be true and trustworthy. For this reason also, science must be freely and openly communicated. My doctoral advisor always emphasized that it does no good to do science if you do not communicate it effectively. The accepted vehicle is the peer-reviewed scientific journal. Peer review is a mechanism to evaluate the quality of experimental design used by an investigator and review the soundness of the results obtained. The importance of the peer in peer review is because peers are the only ones who possess the expertise to understand and evaluate the work. Unfortunately, RTP and other employment pressures have sometimes spawned fake journals that appear legitimate to the outsider, but often have a pay-to-print policy, and in some cases even list prestigious scientists as editors without their knowledge. Publication of scientific findings in peer-reviewed journals of good reputation is the source of scientific truth sharing. Science writing is very different than other types of writing. Fewer words are better, including the sparse use of adjectives and adverbs. Writing is done in the past tense, passive voice. I just saw a posting from a high school teacher that said that she best explains passive voice by asking the students if they can put "by zombies" on the end of every sentence, and then it makes sense. So instead of "I broke the dish was broken by zombies." So, and of course, in my house with 2 scientists, things are always occurring on their own, like dishes breaking and things. Fewer words are better, sparse use. Writing is done in past tense passive voice. One of the most important parts of a journal article is the materials and methods section. And while relatively short, it is often the most difficult to write. It is in this section one must meticulously list all materials or strains of bacteria, for example, used in your work, and then relate the protocol in such detail that other scientists in your field could recreate your results exactly from this information. It is called reproducibility, and it is essential to truth in science. And I would also put a little, depending on the branch of science, for example, in bacterial genetics, it sometimes took us 3 years to build a certain strain just to be able to start the experiments with it. So you have to put all that in there, assuming that somebody will ask you for your strain, and you're obligated to share those. Additionally, there is a deep personal commitment by scientists to maintain their reputation for honesty. In many research circles, you know at least by name all the key figures in a field from around the world. It even goes so far as keeping pedigrees from what research labs that you hail from. In an article published this month in Public Library of Science Biology, PLOS Biology, by John Ioannidis et al. From Stanford University's Meta Research Innovation Center, known by the acronym METRICS, they write about the challenge of evaluating the basic principles of the scientific method when there are over 1 million new research articles published annually. They define meta-research, or research on research, as, quote, an evolving scientific discipline that aims to evaluate and improve research practices. It includes thematic areas of methods, reporting, reproducibility, evaluation, and incentive, close quote. The burgeoning number of publications has spawned the use of impact factors that are meant to represent the importance of a journal by computing this number from the average number of citations received per article published for the preceding 2 years. This is a controversial measurement among some, as there have been incidents of groups ramping up numbers by self-citing. That's illegal in 38 states, I'm pretty sure. Also, some articles represent a very narrow field not commonly cited in scholarly reviews, another major source of high impact factor numbers. How often do published articles stray from the truth? By mistake or by design? A somewhat old but passable editorial from 2011 from the American Society for Microbiology journal, Infection and Immunity, cites the Committee on Publication Ethics survey of Medline, which is a major source of finding articles, Retractions from 1988 to 2004. They found that 40% of retracted articles to be attributed to honest error or non-replicable findings, 28% to research misconduct, 17% to redundant publications, and 17% to redundant publications and 15% to other or unstated reasons. All these aspects of peer-reviewed publication may themselves need to be reviewed in the future if it is expected to function as the true quality control of research. I leave you with a short series of classic experiments that reveal DNA as the hereditary material. And this is a classic example of archival science and publications building upon other publications. In 1928, as all you students know that I make you write about, Frederick Griffith noticed that he could infect mice with a virulent form of streptococcus, and he had an avirulent form of streptococcus that wouldn't make them sick. One made them sick, the other one didn't. And when he took the one that made them sick, the virulent one, and he boiled it and he killed it, it didn't make the mice sick. That makes sense. But when he mixed the dead virulent one with the live, with the live avirulent one, it made the mice sick. So he discovered there was a transforming principle. It was then in 1944 when Avery, McLeod, and McCarty repeated the same experiment, only they used chemicals to select out different biomolecules that could be responsible, that they found DNA was the transforming principle. Finally, in 1952, Hershey Chase radioactively labeled DNA and radioactively labeled protein, and did a little experiment with viruses that infect E. coli. And it's funnily called the wearing blender experiment, because they, to detach the bacteriophage from the bacteria, they poured it in a wearing blender, kitchen blender, and whirred them up. And they found then that the transforming principle was indeed DNA, confirmed again in 1952. And I'm here to tell you that 1952 was 63 years ago. It was a good year. And it was a while ago. And by then, through all these low-tech means, it was known that DNA was the transforming principle. In science, truth evolves, if you forgive the pun. The utility and resilience of the scientific method, along with the requirement of open communication of scientific findings in quality peer-reviewed publications, will keep the ship headed in the right direction, even if it is sometimes deviates from a true course. Thank you for your attention. I have to say, today I was watching the new news, and because I debated about whether or not to talk about untruth in science and truthiness, which is one of my favorite topics lately. And I heard on the new news that there were 10 cases of whooping cough, Bordetella pertussis in Pataskala, totally preventable by vaccination. I also heard that 50% of Americans believe that in communications between the living and the dead and aliens. So we have an obligation to science literacy too. So thank you very much. I'll take any questions if you have any. Dr. Hogarth.
I was wondering what you thought about this statement that some of the tricks that people have played on science generally, the one that comes to mind is Piltdown Man. Do these actually aid the production of a better quality science?
Any of the hoaxes like that?
Becky, could you repeat the
Oh, yeah, what was the name of the man? It was a, yes, a hoax, perpetrated hoaxes on science. And I think they're pretty easily sorted out. There's a cost involved for some of the more serious ones. I think about the false connection between autism and vaccinations, and another one that's cost a huge amount of money is the one about cancer causing high tension lines. And those are classic ones that the harm that they do is that they suck up research money that could be used for something true and valuable. So people spend money to prove or demonstrate the same thing over and over and over because of Jenny McCarthy and that nonsense. So I've seen the figures before, I can't recall them, but they suck up research, badly needed research money. That's the harm in them, I think. So. Any other questions? Yes.
I'm surprised. This state had a big fight just a few years ago about the theory of evolution. Many books were written about it. Many philosophers, many theologians. In fact, at a recent trial, even though all the scientific representatives spoke, absolutely, there's no problems, the agency that was promoting teaching critical critically about evolution, handed over to the court 100 papers, all by major scientists, critical of the theory of evolution. I'm surprised in one sense, but I would think if you were going to give a talk on truth in science, it would be most logical and appropriate to focus on the theory of evolution. So that's a very good topic to talk about. But I'm shocked here in a Catholic university that you can speak about it with no reference to anything ever said critical about it by theologians, philosophers, and scientists.
I hear what you're saying. In fact, I always, many of you have heard this story before, that when I first came here, according to the textbooks and my own information, was teaching this section on evolution. And I asked for, we saw, I don't know, some video or something, and I asked the students to write a reflection about their, what they thought about this, the very thing that you bring up. And I was shocked and surprised when I started getting papers that said, I'm a good Catholic, therefore I don't believe in evolution. Well, I'm not Catholic, and I thought, well, I better get with the program here and find out what's going on. So I contacted our theology department and spoke with Barb Finan and Dr. Leo Madden, and I said, what's going on here? And Dr. Madden said, I'll be glad to come over and talk to your class about evolution and Catholicism, and he did so, and it was very enlightening to the students who thought that the Catholics had something against evolution, which they do not. And he furthermore directed me to the Vatican website that has from their scientific council that again states that there's no problem with evolution according to the Catholic Church. In fact, no major church, no major religious organization has any problem with the theory of evolution. It's not my job to delve into the aspect of life that crosses into the subject areas of religion and theology. My job is to convey the facts, and they are the facts. And everyone is free to believe what they want to believe, but you can't argue with the facts. I have respect for people that believe otherwise. I just happen to think they're wrong. And I think I would be very interested in seeing 100 papers from legitimate scientists that didn't believe in, that didn't support evolution. I'd be glad to look at them. I just don't believe it. Anything else? Thank you. Yes, Ron. Dr. Carstens.
I'm not sure I can ask this question right. And so maybe you can help. It seems to me that pursuit of truth, being a science as you've described it, requires on the part of those doing it an openness to the possibility that there's more than our little categories can exhaust. it seems to me that philosophy and theology consist of the same kind of openness to the possibility and the probability of the way things are. So I wonder if you could just give us your off-the-cuff opinion of why those areas come to loggerheads. I mean, it seems to me that...
No, I hear you. Yeah, I hear you there. I've thought a lot about this, and I've thought a lot about people like Richard Dawkins, for example, and I've had this conversation with Steve Thomas before too. I really think that we all get into trouble when we jump into a pool that isn't our own. I refrain, or I try to refrain from being a philosopher or a theologian and stick to what I know in science, but which by no means limits wherever these other disciplines may take you. I mean, there is, as Sister Maddie said, there is something about science that when you grasp some of these concepts at the level that you end up working on them in a Ph.D., for example, that I have literally heard of people dropping to their knees. And some of being able to grasp I don't even know how to do it. It's without a word to me. That to be able to grasp that this goes on and to just come to the reality of what we know, we truthfully know about science is more than most people can deal with. So I'm always, as I've said to Dr. Thomas, I get very leery when I see science people talking about matters of theology, religion, philosophy. It makes me very nervous. And it's kind of goes into that, falls into that category of beware of people with PhDs after their name being experts on things they don't know a damn thing about, which is, I think, most irritating. I try to be very careful never to speak about something that I don't know about. Or at least when I do, I will give that disclaimer.
But that would then entail a larger community speaking to each other.
I think so. Speaking from my own personal experience, I have often thought that this is so much already. I don't need anything more. I don't need another explanation. It's already enough, with a capital E, I guess you would say, from the old days. Yes?
This isn't very profound, but it's very important to me.
Uh-huh.
What on earth can we do about the cheating in the science classes? These are the people that are going to grow up and do stuff.
I think there's a disturbing trend in general. Like when I mentioned about the journals, nobody 20 years ago would even thought about these fake journals that are out there, that unless you're exactly in that field, you wouldn't know about it. Some of you heard me talk about how there was one journal that was recommended to me about, oh, this is a good place to publish, try this, by a person who wasn't in the field. And I look at it and I go, well, wow, I know that guy, the editor, he's an important guy. We had him as a speaker at Iowa State and everything. And then I look at all the other names and I don't recognize them. And I send him an email and he had no idea he was being listed as the editor of that journal. And it goes, there's psychology people here. There's when I took Ed Psych, I remember them saying that anybody will cheat if enough pressure is put on them. That anybody will do that. Now, I don't know, but that's what I wonder about is, has it become so all-important that we have this rash of dishonesty in the classes as well as in the professional class? I don't know. It's pardon my French, it's a hell of a world where you got to stand over a student and practically videotape them, to keep them from cheating. That's not good. And we have less of that problem here than at the big places where you have to show IDs and they send in ringers. And it's like I say to my students, my only fear is going into an emergency room on a gurney and looking up and seeing one of you. So, not all of you, but some of our problem children. So, okay. Well, thank you very much.